We're now going to talk about the tertiary structure that can occur within a protein. Now, this is after we've potentially formed our hydrogen bonded alpha helixes or our hydrogen bonded beta sheets within a sequence of a peptide and amino acids. But how do we take those now potential helixes or sheets that are existing and really bind them together into one final cohesive three-dimensional structure? This is done through special types of non-covalent bonding and linkages that occur between potentially far points of the whole amino acid peptide structure. Things that hold the tertiary structure of a protein together include ionic bonds. So if we have some type of positively charged residue and some type of negatively charged residue, again, they can be on very far parts of the whole sequence of amino acids. They can actually hold together that portion of the protein and fold it together by having an ionic bond, positive and negative charge bond together. Another main type of bond that occurs that holds together sections of proteins is a disulfide linkage. I brought this up early again in the class when we were talking about functional groups. And I talked about the disulfide bond that can occur. When you have a cysteine amino acid, which remember has an R group that is this SH. So this is the R group of cysteine. Two cysteines can end up forming a covalent bond with each other. So disulfide linkages are special in that they are a covalent bond. Ionic bonds are not covalent. Remember, they are not sharing bonds. They're just a charge interaction that's occurring between positive and negative charge. Similarly, hydrogen bonds are not covalent bonds. They, again, are a sharing of slightly positive and slightly negative charge along a structure. It just so happens that they involve a hydrogen, where the hydrogen is the part that is always partially positive, and the lone pairs on another atom are partially negative. So this is a non-covalent type of bond. This is a non-covalent type of bond. Disulfide linkages are covalent. They are a sharing bond between two sulfur groups, so they are very strong. And these interactions, again, they're very, very tough, strong interactions, and you find them very often within proteins helping to hold together their tertiary structure. The last thing that can help hold together their tertiary structure is hydrophobic interactions. This is not a bond. Remember, we talked about this again in chapter 1. Hydrophobic interactions are it's a interaction that is driven by entropy. So it's just that nonpolar groups like to kind of cluster with other nonpolar groups to get away from water. And this is favorable because then there is less interaction of water, which is polar, with a bunch of nonpolar groups that does not interact with water. So hydrophobic clustering, hydrophobic interactions will occur between nonpolar groups on different parts of the chain. So again, the tertiary structure just refers to this overall arrangement of atoms in the protein. It's stabilized by these weak interactions that I just went over, or it could be by this strong disulfide bond interaction. The covalent bond is strong. These generally help stabilize and form that overall spatial arrangement of the protein. the very final overall 3D structure. And again, these amino acids don't necessarily need to be near each other in that primary structure, in the actual sequence, to help form these interactions. They can be far apart, but the way that they end up interacting helps to fold and kind of cluster together the final structure of the protein. Now, we tend to classify proteins based on this type of tertiary structure they have. And the two classifications are fibrous proteins and globular proteins. And they end up looking like how you would kind of expect them to look. When we talk about fibers, these are usually along strands of some type of structure. So fibrous proteins tend to be in strand formations. Globular proteins usually have a tertiary structure that is, as it sounds, globular. It's a kind of large, almost spherical formation, not in strands. So we're going to go through these two types of protein structures. Again, these are just types of tertiary structure that can exist, how they're held together, and some examples of those tertiary structures. So first is fibrous proteins. Again, fibrous proteins are strand-like types of proteins. They are made up of one type of secondary structure that's then held together in a tertiary structure. So they're either made of entirely alpha helixes or entirely beta sheets, and then those are held together into the single fiber unit of the protein. So here are some examples of that. You can have alpha helixes that, again, a bunch of alpha helixes along the structure of the protein. So think about the secondary structure. Imagine that you have one sequence of amino acids. It's all in alpha helixes, and then those alpha helixes are then kind of bound and interacting together via disulfide bonds. So they're using that tertiary interaction, a disulfide bond, to hold together two different alpha helixes. These types of structures are usually very tough, and they are what make up things like keratin. This is how keratin is structurally made. You can have a beta conformation. This is a lot of beta sheets that are then held together in a final three-dimensional conformation. These tend to be more soft and flexible filaments. So something like silk. Silk is still a fibrous filament, but it is definitely more flexible and soft than something that is made entirely of alpha helixes like keratin, which is very tough. And another example, this is collagen, which again is made of alpha helixes. It actually has a triple helix. We'll look at, again, very high tensile strength. It's very, very tough, right? It's almost bone-like if you think about collagen in your ear. It doesn't really have much stretch. It can hold its shape very well, and it's very tough. So again, this is things that make up like your tendons and bone matrix. So let's look at, again, some examples. Here is keratin. Keratin is a fibrous type protein. It makes the structure of your hair. And essentially it's just an array of lots of alpha helixes. So one keratin protein will essentially form an alpha helix, and then those alpha helixes are bound together via some disulfide linkages, which are then wrapped even more, as you can see in this previous, to form filaments within the cell in your hair. So these proteins end up being, because there's multiple proteins wrapped and wrapped and wrapped and interacting with each other, they end up being very large. They still exist within a cell, but they end up being very, very large, as you can imagine. To hold together the structure of your hair, they have to be pretty large. So you have, again, a protein that is then wrapped around another of the same protein. All of these are just alpha helixes interacting with alpha helixes. And note that I said for this type of alpha helix interaction, they are bound together by these disulfide bridges. So maybe you guys have heard about sulfur in relation to hair, because this is the chemistry of things like permanent waving. So going from straight to curly hair. So you can see when you have these interactions forming disulfide bridges. So this is like an alpha helix and an alpha helix within the keratin. They're interacting in a tertiary structure using a disulfide bond. And that keeps the hair very straight. These disulfide bonds are very rigid, and you end up having very straight hair. Now, if you break these disulfide bonds, and we do this through chemistry, we can reduce the bond. If we put it in a reducing environment, that's the chemical that they essentially put in your hair if you were to get a perm to help get your hair wavy. So if you were to reduce the disulfide bonds, they will break, and you don't have as rigid of structure between these alpha helixes, which causes them to kind of mismatch. And then lastly, we reoxidize them. So once they've now kind of mismatched with each other, they're not in this straight disulfide linkage, they've formed out of interaction between the alpha helixes. We then reoxidize, reform any disulfide bonds that are near each other. But because these have now misplaced, the alpha helixes aren't exactly next to each other in their positioning, you will have a now locked in curl because you have less rigid and lined up interactions between your two keratin strands. And that's how you get a perm. So you can also imagine how chemical interactions could help with straightening of hair. So think about that and maybe come to me in our meeting class and we'll talk about how you chemically straighten hair based on these properties you just saw. Okay, so collagen again is another example of this type of fibrous protein. Collagen forms very important parts of our tissue, tendons, cartilage, bones, even the corneas of your eyes are formed by the collagen protein. Collagen, each chain is just a very long alpha helix chain of glycine and proline rich left-handed helixes. So it has a left-handed structure, and the R groups on this are very rich in the glycine and proline. These collagen chains then intertwine with each other into a triple helix, and this triple helix is actually right-handed in nature. So when they intertwine with each other, the direction of the winding of the three chains together is a right-handed helix. This triple helix ends up having more strength than things like steel wire that is the same diameter. So protein interactions can be very strong. Here's just an image of a collagen fiber if you were to look at a cross section of it. And again, this is the triple helix that you're seeing in this. So this is the right-handed triple helix, the three chains that have then wound together. And this is what a sectioning kind of imagination of what each of the alpha helixes would look like within the triple helix. This triple helix is bound together again by crosslinks that are usually bonds between lysine or histidine amino acid residues. The last example we'll give for a fibrous protein is one that utilizes beta sheets instead of alpha helixes, and that is spider silk. So spider silk can be used for webs and spider egg sacs, and even, of course, wrapping of prey. So it has to be very stretchy and versatile, but it also still has to be very strong. Again, this material is stronger than steel at the same diameter, but it can stretch a lot before breaking, and that the ability of it to stretch has to do with the fact that it's using these less tense beta sheet formation instead of those alpha helice formations. Here you can see this is 3 beta sheets, and you can see each of these is a sheet, right? It has a strand going back and forth, back and forth, back and forth. And then here's another whole sheet and another whole sheet. So again, this is a fibrous protein, which is protein that uses a secondary structure, repeat of a secondary structure to build its whole final three-dimensional structure. These beta sheets tend to be rich in alanine and glycine side chains to hold together through hydrophobic interactions the sheets, each of the sheets on top of each other. Okay, so now we're going to get into the globular proteins, which is our other type of tertiary structured protein. We talked about fibrous proteins, again, which are proteins that have repeating secondary structure, the same type of secondary structure that is then held together by some type of interactions, these tertiary interactions. Here we'll discuss globular proteins, which have multiple types of secondary structure within one protein. They tend to not have a very structured form in that they don't have, repeating units. They don't have a singular strand-like structure. They tend to be more spherical in nature in their final three-dimensional look. These types of globular proteins can have an arrangement of several of those secondary structures we discussed. So they can have alpha helixes, beta sheets, or they can have both. They can still have all alpha helixes, but they tend to not be in an arrangement in which they are fibrous in nature. So they might have a kind of alpha helix here and there, that's all they have, or they have a beta sheet here and there, but not these repeating alpha helixes or repeating beta sheets that are then stacked upon each other. So again, they can have all of them, all alpha, all beta, technically. They can have both as well. Generally, they have a mix of the two of them. Now, these globular proteins are comprised of what we call motifs. This is just a fancy word to say a type of recognized fold, a type of fold we see very often that is in multiple types of protein structures made up of these secondary alpha helix or beta sheet structures. So some examples of those motifs. Again, these are just common types of structure you find within a globular protein. A beta alpha beta loop. So this is a portion, a segment of a protein that has a beta strand followed by an alpha helix and then another beta strand kind of interacting with the very first beta strand. This type of loop, the beta alpha beta loop, again, common motif you see in globular proteins. Another one is a beta barrel. This is something that's made of only beta sheets. Again, usually you see these as only small segments. They don't make up the entire protein, but will be a segment of the protein that forms this barrel-like structure of sheets that are wrapped together. Another example is an alpha beta barrel. So this is this kind of barrel-like rounded structure that's made up of multiple portions of the peptide that are beta sheets interacting within kind of the central region, with the outside region being alpha helices. And remember that these alpha helices and beta sheet portions of the strand can kind of occur at any point along that sequence of amino acids. There's also portions of globular proteins that we call intrinsically disordered. Um, this is any type of protein segment that just lacks any definable structure. So it doesn't have any definable alpha helix or beta sheet secondary structure. It just is kind of, again, disordered. It's the amino acids. Maybe they're held together by one or two um, non-covalent bonds, but generally they don't have any structure or order to them. So proteins can have either segments of this intrinsic disorder, or the protein can have large portions of it that is intrinsically disordered. Again, this happens only in the globular-like proteins. And generally, when you have a composition of amino acids that are going to break structure, and you have a higher composition of them, like lysine, arginine, glutamate, or proline, these ones tend to kind of generally break the definable structure of a protein. And then you tend to have more disordered region. Disordered regions aren't bad. I'm talking about them, but they're not a negative thing. Having an area with less order to it can actually be good for a protein. And that's because they're more malleable. They have the ability to move within the space of a cell, for example, or in a lipid membrane. And they actually can help facilitate interactions with other proteins. So these disordered regions, these flexible regions, sometimes benefit the protein in whatever the type of interaction that it ends up having.